This is Unit 8, Part 2. We covered earlier in Part 1, we were talking about transcription and translation in prokaryotes and eukaryotic cells. Now we'll talk about mutations. Now mutations occur all the time. Mutations are a normal occurrence that happens to DNA. It happens because of errors in copying of the DNA going from generation to generation. You get an error in the DNA replication, and there are agents that can cause mutations or increase the frequency of mutations, but mutations would occur anyhow. Spontaneous mutations occur in any case, even in the absence of mutagen. The key thing to understanding mutants and mutations is that they are not always harmful at all. Quite often they have no effect, they're neutral, and they can indeed be beneficial at times. Okay, now mutations can occur during DNA replication. The DNA undergoes replication just before the cell, if it's a eukaryotic cell, just before the cell undergoes mitosis, or if it's a prokaryotic cell of bacteria, just before it undergoes binary fission. The alteration of a single nucleotide in the DNA coding strand in a gene can have important effects. In the normal situation, I remind you that when a gene in the DNA, genes are in a single strand of the double-stranded DNA, so the gene would be on the coding strand. By the process of transcription, a messenger RNA transcript is made. So transcription leads to a messenger RNA transcript, and then that information in the transcript is used to make the particular protein. And as you know, the information is arranged in codons, 3 nucleotide long, and if there was a mistake, then you can get an error in a single nucleotide that changes things fairly dramatically, at least in terms of, if it's a single nucleotide, it can change the one of the amino acids because it changes a codon. Now, in the next slide, we have an example of an alteration in a codon. Just keep your eye on this here on the next slide. And that C was changed to a T due to a mutation when this particular DNA was copied or replicated, and that results in a change in the single amino acid. So this is called a missense mutation, where you have a change in an amino acid, and the primary structure of a protein, in other words, the amino acid sequence, I remind you that primary structure refers to the amino acid sequence, can in fact have quite a significant effect on the shape of the overall molecule, and therefore can actually change the function. Now, it may increase the function, or decrease function, or occasionally have no effect, but the point is that the mutations that do end up with, due to a single base pair change, that end up with a change in amino acid, are called missense mutations, and can actually be cause a fairly important change. Go back to the example of the normal transcript and translation. And here it was glycine, and that got changed in that missense translation on the previous slide into a serine. Now, there are other types of mutations. For example, there are nonsense mutations. In this case, you again get a base substitution. Base substitutions are called point mutations, and this sentence actually should have been on that previous slide that showed missense mutation, because it was also a base substitution. one nucleotide base was substituted in that one. Here it's a nonsense mutation resulting in a nonsense codon. You get a stop codon, for example, here that immediately stops the process of translation of the message, and so the product here in this example is complete nonsense. It's a single amino acid, and it stops. So that's not going to have the whole normally produced peptide is not produced at all. So that's a nonsense. Another type of mutation that can change things, and if codons are in groups of 3, then and one of the nucleotides is deleted or added, then what happens is you get a frame shift mutation. So a frame shift mutation means that the reading frame is changed. Here you have the start signal codon, then the next codon, and one of the nucleotides was deleted in that mutational event. So you get a shift in the frame. Instead of AAA giving you UU, a codon UUU, the whole frame is moved over one nucleotide, and so you get a change in that amino acid that's coded for, and in the next, etc., all the way down. And these 3 dots means it continues onwards, and it'll continue onwards until you hit a stop codon. So this is a frame shift mutation, and it could be 1 or it says more. If it's 1 or 2 nucleotides, then you have a frame shift mutation. If it's 3, you get a drop out of a single codon. So it's of course it depends where the 3 are, but it can be 1 or more nucleotide pairs that get inserted or deleted, causing a frame shift. So we've talked about base substitutions resulting in point mutations, in missense or nonsense mutations, and here we're talking about insertion or deletion as opposed to a change in nucleotide. An insertion of deletion or more of 1 or more nucleotides, and therefore it's a frame shift mutation. These kind of mutations do occur, as I said, spontaneously, and in eukaryotic organisms, bacteria, for any single gene, on average, typically mutations occur. The frequency is about a 1 in a million chance, or mutation rate the probability of a mutation in any single gene is about 1 in a million. So when the bacteria undergoes DNA replication, is going to divide, there's a 1 in a million chance that there will be a mutation in any one particular gene. So if you look at 1 million bacteria in the next generation, one of them will have a particular mutation in a particular gene. This mutation frequency may sound small, but seeing as though bacteria can expand in numbers very, very rapidly, and in 24 hours you can go from a very small number to millions and millions of them, then you should realize that it's frequent enough that it is significant. There are repair mechanisms that can repair some mutations, but nevertheless the mutations will randomly occur throughout the chromosome at a 1 in a million probability of happening. They occur more frequently, but there are repair mechanisms that can repair mutations, but they don't catch all mutations. So it's important to understand the mutations, in fact, are part of the process of the natural process of evolution. That is how we evolve. That is how new species or mutations, changes in species, changes in genes come about. The mutation resulting in antibiotic resistance, for example, in this case it's advantageous to the bacterium. because it gives it a growth advantage if there is antibiotic present, and so that particular mutant organism would become more frequent if antibiotics are being used and there is a single one that has antibiotic resistance. Then in a fairly short time, quite quickly, it will become its representation in the overall population, the frequency of mutants in the population will rise tremendously, because those are the ones that have a survival advantage. Well, we can study mutations and detect mutations fairly easily in bacteria using a fairly simple approach, where you have culture that has, if you're growing a single type of bacterium and you have individual colonies on a plate, then you can just take a sterile velvet pad on a setup like this and gently press it onto the surface, and what you end up with is samples of the colony on that pad, and then you can make replicate plates, and you know exactly the orientation by having marked all of these carefully with an X here and X there, and you can see that this is marked and this is marked, so you know, and this is marked here, so you know exactly where or how the orientation is and which colony is which, and you get an exact duplicate. In this particular plate, in this particular example, and it doesn't matter what the mutation is, but here we're looking at a particular one, but this can be done with all kinds of different mutations. In this case, they're looking at mutations in a gene which allows the organism to in this case, we're looking at a gene that allows the organism to in this example, we're looking at a mutation in a gene which allows an organism to grow in the absence of histidine. So if it cannot grow in medium lacking histidine, it has a particular mutation. So all of these need to be growing in medium containing histidine. You see, all the colonies, but there is in fact one colony missing on this plate. This plate does not have histidine, and so there was a mutation in the gene that makes the organism require histidine. Okay, so that particular bacterium is represented by this colony here on this plate. These two plates were plated using replicate plating, and one colony is missing on the plate with medium lacking histidine. So these bacteria in this colony here have a mutation in the gene that allows them to grow in the absence of histidine. Okay, so we're looking at a change in colonies, a missing colony. There's all kinds of theoretical approaches. This one's, I guess, a little confusing because it's a double negative, not grow without histidine, but that is a strategy in demonstrating mutation. Okay, there are agents, as I told you, that increase the frequency of mutation. They are called mutagens. They can be chemicals or energy, high energy, exposure to higher energy like X-rays or ultraviolet irradiation that can damage DNA. These have potentially mutagenic effects, and they increase the mutation frequency 10 to 1,000-fold. So instead of being 1 in a million, it can be 1 in 100,000 or as much as 1 in 1,000. The test that is classically used to see if a material does have mutagenic effects is called the Ames test. So anything that is going to be ingested, any new product that is going to be eaten or drunk or used as a drug, taken internally or used on the surface of the body, will need to be tested in the Ames test. It's a classic test looking to see if there's an increase in mutation frequency, and they use bacteria to see if mutations increase in the presence of the product that they're testing. And the experimental approach is fairly simple, and again the example here is medium lacking histidine. Is there if there is growth or isn't growth with or without the histidine, if there is mutation in the histidine requiring growth of these bacteria. Okay, now let's look at transfer of genetic material between organisms. Okay, we talked a little bit actually about that before when we talked about transduction. We were talking about bacteriophages, which are viruses that infect bacteria, and I gave you an example in that lecture. I think it was lecture 7 on special. It was actually specialized transduction, where you have the prophage DNA inserted into the host bacterial cell's genome. When it came out and it started going into the lytic phase, it came out but took with it a little bit of the host cell DNA that was located right beside the prophage DNA. That's called specialized transduction, and can result in transfer of a bacterial host cell DNA into another bacteria, because if it gets packaged into a bacteriophage and then that bacteriophage infects another bacterial cell that lacks the gene that was located beside the prophage, then it's transferring over that genetic material. The kind of transfer, any transfer of genetic information, genetic material between organisms, is called recombination. So it's a question of recombining of DNA that was originally from more than one organism. Genetic recombination, in fact, can be an exchange or transfer of genes. There are two major types of transfer. One is vertical and gene transfer, and the other one is horizontal. Vertical refers to transfer going from one generation to the next generation, to the daughter cells that are produced when you have DNA replication and binary fission in bacteria, or in humans, in eukaryotic cells, it would be transfer from cell to it would be DNA replication before mitosis. But here it's all within the from one generation. Here we're talking about bacteria, and it's from one generation to the next of bacteria. So from the parent to the two daughter cells in the next generation. That is vertical gene transfer that can occur. But the other that occurs potentially is horizontal gene transfer, where a gene is transferred between cells that are alive at the same time and within the same generation. One cell that gives the genetic material or gene is called the donor cell, and the other one's the recipient cell. And there's several ways that can occur. The first type of horizontal transfer that I'll talk about is a very simple one that was actually used to prove that DNA was in fact the source of genetic information. When they were first trying to figure out where genetic information was kept in organisms, they were thinking, some people were thinking it was in protein, and some were thinking it was in DNA. There were many proposals, but in the end, this kind of experiment that I'm going to show on the next slide proved that it was in the DNA actually. So this is called transformation, in which genes are transferred as free, naked DNA. Bacteria do, not all, but many bacteria do have the ability to take up free DNA. The example that I'm going to talk about is in Streptococcus pneumonia. There is a gene which can code for capsule. Streptococcus pneumonia organisms that have a capsule are more pathogenic because they are resistant to phagocytosis by phagocytic cells like macrophages and neutrophils, which not only phagocytose but kill the bacteria. Those genes, those that have the gene for the capsule are more resistant and therefore more pathogenic. It's easier for them to grow in an animal, infect an animal, and successfully grow. Some don't have that gene and some do. Now, what they did was they took live encapsulated organisms, injected into a mouse, and the mouse died, and then they were able to isolate and grow colonies that did have the capsule. If they injected a mouse with unencapsulated bacteria, then the mouse didn't even get sick, remained healthy, and they are able to isolate and grow colonies of bacteria that are non-encapsulated. If they, in a third group of animals, if they killed heat-killed encapsulated bacteria and injected that material into the mouse, then the mouse remained healthy and no colonies of any of the bacteria could be isolated. No types of bacteria could be isolated and grown from the mouse. If they mixed living, non-encapsulated with heat-killed encapsulated bacteria and injected that mixture, and remember the encapsulated ones are heat-killed in this case. So these are heat-killed. The two are mixed together for a short period of time and then injected. What happens is the mice get sick and die. And colonies of bacteria can be grown from the dead animal, and that they are encapsulated bacteria. And the explanation for this was, in fact, that the free DNA, the DNA that is freed when the organisms were heat-killed, was taken up by the unencapsulated, and so then they had the ability to express a capsule. So genes can be transferred by this process, horizontally transferred by this process of transformation. And here you see an example of recombination by transformation shown at the single cell bacteria level, where you have some free DNA here, DNA fragments that are taken up and inserted into the bacterial genome. Now, another type of horizontal gene transfer is called conjugation, and this is different. In this case, bacteria do not only contain a single chromosome, double-stranded circular chromosome, but they also quite often contain small circular pieces of double-stranded DNA called plasmids. These plasmids can code for a few genes, and plasmids are self-replicating. So if you have a bacterium that contains a plasmid and it undergoes binary fission, you get the next generation. The plasmid can be transferred with that into the next generation. So you have, of course, the host bacterial chromosome, which is much, much bigger than a plasmid, but you can also have plasmids inside, usually more than one. And the daughter bacteria will have the two second generation bacteria will also contain the same plasmid. It gets transferred through the generations because the plasmid is self-replicating. So you have that it's in the population and it stays in the population with the population as the population of bacteria grow. However, what is important in terms of horizontal, and that would be an example of a vertical vertical transfer of a gene, but what can also happen is that some bacteria are able to form what's called a conjugation tube. And plasmids can be exchanged through, there's a conjugation tube there. I don't know if you can see there, that's a conjugation tube formed between the cells. And plasmids can be exchanged from one bacterium into the next through these kinds of conjugation tubes. And once it's exchanged, because the plasmid is self-replicating, it will, get more than one copy, and it will continue on as in the horizontally transferred, vertically also transferred as the bacteria that received it divides and grows. So that's an example, a second example of horizontal gene transfer. The first was transformation, the second is conjugation, and here is a diagram showing that process of conjugation. And the third that I want to talk about is transduction, another type of horizontal gene transfer where it's transferred by the bacteriophage. The example I talked about in lecture 7 was specialized transduction, where it's a transfer by bacteriophages when they pick up a piece of DNA that's immediately adjacent to prophage DNA inserted in the genome. In this case, this is called generalized transduction, where the host chromosome breaks up as the host cell dies in a lytic type of life cycle of bacteriophage infection, and you have the host chromosome breaking up and some host chromosome getting packaged into new phage protein coat, new phage particles, and that new particle with the host cell DNA in it infecting another bacterium. So here you see this kind of situation where you have a bacteriophage infecting a bacteria, and they've left out here in this second part, they've left out the fact that the phage DNA and they talk about it, but they don't show it in the diagram, but new phage is being made and new phage DNA is being made, but there's also lysis of the host cell DNA, and some of it gets packaged, and it's shown here, for example, and here. Some gets packaged in bacteriophage, and then that phage goes on to infect a new bacteria, and so the previous host's DNA can, in fact, if it's a lysogenic kind of cycle, then it can get inserted into the bacterial prophage, and this is generalized transduction. I wouldn't worry about specialized or generalized. Know and understand transduction. So those are three types of horizontal transfer: transformation, naked DNA, conjugation, transfer of plasmid, and here transduction through the action of a bacteriophage. Okay. So those plasmids that I talked about and told you were self-replicating, much smaller than the circular chromosome, the single chromosome found in bacteria, but they are self-replicating small circular pieces of DNA found in bacteria. They can contain a small number of genes, and sometimes those genes increase pathogenicity of organisms. For example, E. coli infections, there are many subvariant types of E. coli, Escherichia coli, and some have toxins that cause what's called traveler's diarrhea or infant diarrhea. And that toxin is typically expressed in a gene within a plasmid. Clostridium tetani contains a neurotoxin. Clostridium tetani is the organism that causes tetanus, the disease tetanus, and it is due to a neurotoxin. And that neurotoxin also is expressed in a gene in plasmid. Bacillus anthracis is the organism that causes the disease anthrax, and the toxins associated with that, that pathogenicity or pathogenic effect of the organism, the process of the disease anthrax, are also expressed in plasmids. So here you see a photoelectron micrograph of some plasmids. They are quite tiny. And here you see two maps of the same plasmid. This is the diagram. The top one, A, is the diagram in your textbook, and this is the B here is from an earlier edition of your textbook. The map of this chromosome shows it's a double-stranded, plasmids are double-stranded DNA, and it shows different kinds of resistant gene expressed. These are not that first one, but the others are antibiotic resistant genes. You can imagine that conjugation of a bacteria resulting in the transfer of a plasmid that has antibiotic resistance can be quite serious, because a harmless bacterium that has, for example, streptomycin resistance could transfer that to a pathogenic bacteria that causes a disease that normally is treated with streptomycin and now is resistant. The reason I show the diagram from an older edition of the text is because it shows the proper identification of the genes. Genes are identified by using italicized three-letter abbreviated names. So this one, sulfonamide resistance, this one here is written as SUL. Streptomycin is written as STR. Chloramphenicol resistance here, CML. You see? Tetracycline, TET, right down there. Okay, so we've talked about transfer of genes, and these were natural processes. Transformation, the horizontal one, well, vertical is obviously, vertical gene transfer is obviously natural, but also the other three types of horizontal gene transfer is natural processes. Transformation, conjugation, and transduction are all naturally occurring processes that routinely occur. But once these kind of mechanisms were understood, and several other naturally occurring mechanisms were understood, scientists started to use them as tools to design useful tools and come up with methods to be able to transfer genes from one organism to another, transfer a gene of interest from one organism to another so it could be expressed in that other organism. And what we have today, for example, or just to take one example, there are hundreds, if not thousands, no, there are thousands, if not tens of thousands of recombinant genes that have been transferred and are useful and are produced. Just to give one example, the gene coding for the human protein erythropoietin, and I remind you that erythropoietin is a growth factor for red blood cells, was the gene was identified, isolated, and transferred into eukaryotic cells that can be grown in culture, and these are produced. These are grown commercially. They secrete erythropoietin into the growth medium. These are grown in gigantic vessel tanks, reactor tanks. They secrete the erythropoietin protein, human erythropoietin, and it's purified and processed and manufactured and sold out and injected into many people. There's probably no way that you would work in a hospital as a nurse and not be injecting someone within your first week or two with injecting people with erythropoietin. It's such a commonly used medication now. So the recombinant DNA technology simply refers to the transfer of a gene from one organism into the cell of some other organism. And we can use, though, that process to our advantage. Let's look at some of the techniques that are used. The biotechnology, of course, uses microorganisms or cells or components of cells to make a product, and the product can be all kinds of things, actually. Recombinant DNA technology, as I said, simply refers to transfer of genes between organisms. Genes are getting recombined. Genes from one organism is recombined with the genes from another. It's an insertion or modification of a gene to produce a desired product, protein. Genes code for proteins. And here again we have the example of this plasmid. These plasmids are very useful as transfer vessels, which are called vectors. They are vectors that can be used to carry a gene from one organism to the next. They can be designed so that they are useful in that process. They can be designed. They are designed. There are many different kinds now that are available. That they can be opened up. A gene can be inserted. They can be closed up again and then transferred over to another organism. So here we see, and this is a simple example of transfer of genes using plasmids. Plasmids are used for transfer of genes from one bacteria to the next, but now there are processes, more sophisticated vectors that allow us to transfer genes from a eukaryotic cell into another eukaryotic cell. But this is the first type that was used and is still used, and I'll just go through this. Here is a very small abbreviated diagram showing a much smaller than actual size of bacterial chromosome. Plasmids are very small compared to the chromosome, and the plasmid can act as a vector. It can be isolated and opened up, and a gene can be inserted. So if you take DNA from humans and chop it up using specific enzymes, so get all these pieces, then they can be inserted into a vector, a plasmid vector, and the plasmid can then be inserted into bacteria. So the cells there will be bacterial cells with the gene of interest, and they'll start to produce and by gene of interest, I mean, as in the example I talked about earlier, for example, the human gene that codes for erythropoietin, but there are thousands of different ones that are of interest and transfers have been done. So once that gene is in the bacteria, then you can start growing it in large quantities, and they produce the protein, and the protein can be purified from the culture medium and processed and used as a drug. And the kind of things that are done, just to give you a small example, just a very few, human insulin, human growth hormone. The hepatitis B vaccine is produced by this process. It is produced in a yeast. This happens to be the same yeast that is used for wine and beer production, but it has had the gene coding for a particular product from hepatitis B virus. It's a protein that's found on the surface of the hepatitis B virus, and the gene was transferred into this yeast, and in fact produced in huge quantity. And in fact, all of you have been vaccinated with it. It's called the hepatitis B surface antigen. There are other proteins, as I mentioned earlier, erythropoietin, HPV, the human papillomavirus vaccine. The viral proteins have also been produced in Saccharomyces cerevisiae, that same yeast that I mentioned down here, and is used to vaccinate people against HPV, the types of HPV that are known to cause cervical cancer and thus prevent cancer of the cervix. So wonderful, wonderful things can be done using this technology. The sky's the limit. We've only scratched the surface so far. It's only been around now for about, well, it's been around for about 40 years, but used in large, on an industrial scale only for about 25 to 30. More examples: influenza vaccine, well, all kinds of things, all kinds of proteins. Now, I wanted to talk to you about these particular kind of enzymes. They're called restriction enzymes. They occur naturally in bacteria. Bacteria use them to defend against bacteriophage viral infections. They will destroy the bacteriophage DNA that gets put into the bacteria during the penetration phase of the bacteriophage infection. I remind you that during penetration, what happens is the bacteriophage that landed on the surface of a bacteria, the virus that landed on the surface of the bacteria, what it does is I'll draw in another color, it injects its DNA into the infected cell. And many bacteria actually have these enzymes, these restriction enzymes that can chop up, that can cut up the bacteriophage DNA that gets injected in. But enzymes, as you know or should realize, each different enzyme has a very specific substrate that it acts on, and restriction enzymes are no different, and many have now been identified and are produced in large quantities, and they can extremely precisely cut DNA. Many of them typically cut the DNA in a 6 base pair length of the DNA, and it is so incredibly precise that they will cut between particular types of nucleotides, only between certain nucleotides within certain types of 6 base pair sequences. So for example, this one cuts between GG in the sequence GGATCC. If you notice GGATCC, if you look at the complementary strand, so this is GGATCC, the complementary strand is the same but in reverse. These are called palindromes. A palindrome is a sequence that reads the same way in both directions. Palindrome. Okay. There are famous short phrases that are palindromic. Able was I ere I saw Elba. See nurses run. those read the same way forwards as they read backwards. So 6 base pair long DNA segments will be recognized and cut at particular positions. This is another example. The second one is EcoR1, the restriction enzyme EcoR1. EcoR1 isolated from E. coli. The recognition site is GAATC. GAATTC. It's a palindromic sequence because on the complementary strand reads the same way backwards. You see it here, GAATTC read that way, and GAATTC read this way, and the cut is between the G and the A. So you get a cut right there and there. And so you get separation, opening up of the DNA that's treated with this particular restriction enzyme. End result is that you get opening up. This is the example here is using EcoR1 restriction enzyme. Restriction sites again, GAATTC, GAATTC, as you can see right here. GAATTC, GAATTC, and it opens up and you get these overhanging single-stranded pieces which can now combine with another complementary strand. Now you should understand, hopefully, that if DNA in a plasmid is cut using a particular restriction enzyme, and then combined with small pieces of DNA from, this is a bacterial plasmid, say you took some human DNA and chopped it up with the same enzyme, because it was cut with the same enzyme, it will have complementarily the same available strands and it could insert. And here you see an insertion occurring here because it was cut with the same restriction enzyme, and so it can insert right in, and that's how DNA is recombined into a plasma vector, and then a plasmid vector, and then the vector can be put into DNA, and in that way DNA from one organism can be transferred into another. So these are useful tools based on the fact that bacteria were recognized to have these plasmids, and also that it was understood that some bacteria also had restriction enzymes, and that restriction enzymes could be used as tools to cut open plasmids. Here again as examples, we just looked at EcoR1, the use of EcoR1 to open up a plasmid. I hope you noticed that the plasmid was in fact cut at 2 points. There were 2 EcoR1 restriction or recognition sites. So it was cut, and then DNA from another organism that had been cut with the same I'm just going to get rid of this. DNA that had been cut, DNA from another organism, a segment of DNA from another organism that had been cut with the same enzymes was able to be inserted in just by mixing the two together, just by virtue of the fact that they have these sticky ends of CTTA or AATTC, and both have the complementary sticky ends, and they'll spontaneously attach and insert. Here again you have the example, and you see this AATTC AATT single strand overhang. This overhang, and it will combine with this overhang here. You see here and here. They are combining. AATT. Those two areas, and because it's both sides, the other side also here and here, you get insertion into the vector, and therefore the ability to transfer over these pieces into another organism.